Welcome to Gradcast, the official podcast of the Society of Graduate Students at the University of Western Ontario. Coming to you from the other London, let's start the show.
I'm your host, Alex Moszczynski, and I'm joined by my co-host, Jina today. How are you today, Jina?
I'm very good. How are you?
Good. I'm actually great because it's my first time hosting live on air with you again since you got back.
Woo!
And when did you leave for the summer? It was like late June or?
Yeah. So I left early July to go to Korea for the summer for about a month and a bit. So that was great, but I'm back now.
We missed you here, so welcome back.
Thank you.
How was Korea? What was it like over there while you were there?
Korea was very hot and humid. It was not like the Canadian weather that we have here.
Now, was it worse than London humidity? Because we're both from Toronto.
Yes.
And when I came to London, I don't know about you, but I found it super humid and muggy here in the summers. So is it worse in Korea? Is like next level?
It's really no, it's so much worse than London.
So I'll only ever go to Korea in the winter.
Oh, yeah, that's a good idea.
All right. Well, it's nice to be back on air with you. So we're joined today by our guest, Jacqueline Dron. Said it right. And how are you today, Jacqueline?
I'm pretty good, for a Tuesday, I guess. It's Tuesday. Yeah, it's Tuesday.
Tuesday's a great day of the week because Gradcast is on. So tell us a little bit about yourself. So you're a first-year master's student, just started, less than a month in, and you're supervised by Dr. Rob Hegley. All right, so what do you guys do in your lab?
Well, it depends on who you're asking. I mean, there's a lot of stuff that goes on in my lab. So my supervisor, he's a clinician, so he actually has patients that he sees, every I think he has clinic a couple times a week. So lots of patients, and so there's a lot of kind of clinical stuff that we do. So when the patients come in, they might need some sort of a genetic test regarding their particular condition. So my supervisor, he works with lipid patients and things like that. So there's a lot of genetic tests and screens that go on, and I'm sure there's lots of other stuff that goes on in the lab. I just haven't figured it out yet, but really there's a lot going on. So if you get someone else from the lab, they could probably tell you a different story.
Okay. And so I guess you've just started. Why did you choose this lab?
Well, I volunteered at this lab for during my undergrad, and so I was familiar with the people. It was a really great environment. I liked what they were doing. So it's a human genetics lab, and my undergrad was in genetics. So I was really interested in what they were doing. I liked everyone there. I knew already that it was, a well set up lab, and I felt very comfortable. So I figured, well, why not continue here if it's such a good fit?
I would completely agree with you. I've talked to a lot of people, and it seems like a big consensus is that it really matters like how you feel around the people you're working with in your lab, and I guess anywhere you're working. So that's really cool. I guess this makes my next question a little bit moot, but I'm going to ask it anyway. How did you find the transition from like your undergrad to graduate studies? And you're sort of in that transitionary phase right now, but I know you've been working there over the summer as well. So how have you found life?
Well, the environment itself, it was an easy transition because, again, I knew everyone there. But in terms of, actual academic stuff, so it's kind of bizarre thinking that I only, have one class for 6 weeks, and I don't have, a class schedule where I have to be on campus for, X hours a day. So that's been interesting. So far, it's been so good. I haven't, missed running from, health sci to NCB and all those silly things. But I don't know. I'll wonder once midterm or undergrad midterm season kind of comes along, I wonder if I'll feel weird not having midterms. I don't know. So far it's been okay, but we'll see as time progresses.
I find the really weird question is when people find out that like I'm a student still, they'll say, so like, what are your classes like? Or, oh, are you looking forward to the summer? And it's super awkward sometimes to tell someone like at the bank or a stranger that I'm a student all the time, but I don't have class. It's kind of weird. Okay, so you've touched on this a little bit, but let's get into it a little bit more. What subject do you specifically study? So like what's the kind of focus of your project?
So my project is focusing more on cardiovascular genetics and more specifically on a particular type of it's called the lipoproteins. So I'm studying high-density lipoproteins and the cholesterol that's associated with them. So in layman's terms, what a lipoprotein is, it's kind of like a dump truck that just kind of shuttles cholesterol throughout the body. So that's very simply what HDL is. And so I'm looking at different genetic variants or mutations, things at the genetic level that can influence an individual's level of HDL cholesterol. And that's typically the good cholesterol that you hear about. You have good and bad, so it's the good cholesterol that I'm studying.
So you hear about good and bad cholesterol, and there's HDL, and then the other one is low-density lipoprotein, or LDL. Can you tell us a little bit about the differences between the two, and maybe why one is good or another might be bad?
Yeah, sure. So high-density lipoproteins, or HDL, the good cholesterol. So what it does is you can think about it as moving cholesterol from your arterial walls into the liver, so the cholesterol can be removed from your body. So why this is important is because when people have a high, have a lot of cholesterol developing or building up in their arterial walls, you can develop atherosclerosis and have heart attacks and things like that. So that's why HDL is good, because it removes the cholesterol from those areas. Whereas LDL, which is the bad cholesterol, it trucks the cholesterol into your arterial walls. So that's why if you have a lot of, the bad cholesterol, that means you have a lot of cholesterol in your arterial walls. So that's kind of the paradigm between the two. One's taking it to, and one is taking it away.
If I might ask one question, sorry. So then what's the evolutionary advantage here? So when did it become useful for something to say, hey, you got this extra fat, you know where it belongs? Your blood vessels. That seems like a good place to put it. How did that manage natural selection?
Well, there is a biological reason for having cholesterol move to your arterial walls, and it's a little more complex than what I think I can be able to explain. But, your cells need cholesterol for certain things to, synthesize certain materials and things like that. So cholesterol is needed. It's a building block, but it's just when, too much of the cholesterol is deposited, that's when things get a little rough. But there's good to it, but, too much is a bad thing.
Okay, that's awesome. So you're looking at HDL and LDL. How are you looking at, I guess not LDL, you're looking at HDL specifically, but how are you assessing it? What are you looking at exactly that has to do with HDL?
Well, within my project, we have a certain group of patients who have either really high levels of HDL cholesterol or really low levels. And so these individuals are interesting because we're not sure why they have their specific level of HDL. So there can, some people have, really low levels of HDL because they don't exercise regularly, they eat, kind of crappy foods and things like that. But these individuals, they have a relatively normal kind of lifestyle, but they still have these extreme levels. So what I'm doing is I'm looking at the genetic level to see if they have a particular mutation that can explain why they have such extreme levels of HDL. So I'm trying to figure out that genetic side of things.
Okay. So, hypothetical question. Let's say a person eats really, really, really badly and goes to the gym a lot. Are they at risk of having bad cholesterol, or what would you think might be the case there?
Well, I mean, it's really hard because cholesterol levels, it's a very complex trait. So, you have your environmental factors that can influence it. But since it's also, has some sort of genetic players involved, it's really hard to say because, some people might have, they smoke, they don't exercise, all those bad things, but they might have a mutation that is really positive for their cholesterol. So, they could do whatever they want and still not have a heart attack, whereas the opposite is true. So it's really hard to say on just a general example. It could really go either way.
So I just gave a presentation on fat cells, adipocytes. So I'm wondering if you can make that relationship between fat cells and the lipoprotein that you study.
Oh, man.
I guess we have, to my understanding, we have two types of fat cells. We have the white fat, which is considered bad, and the brown fat, which is considered good. So do you think that relates to the level of the lipoprotein?
See, that's a really great question. So at this point, from, the amount I've done for my master's so far, I haven't got to those types of, the fat tissues and everything. So I can't confidently say anything on that. But I mean, I know everything kind of comes together, and HDL cholesterol can come from multiple sources. So I know that these fat cells do play a role. At this point, I just don't know how much of a role they play.
So what you're doing though is more of a genetic analysis. So are there like cholesterol genes, or like how is cholesterol regulated? Do we have like genes that are saying, no, I'm going to have this much cholesterol, or is it genes that are indirectly relating to it? What kinds of genes are you looking at?
There's actually quite a few. So just with HDL in particular, I think there are 7 genes that are there are a lot of genes related to HDL, but 7 in particular that really have a big influence on your HDL levels. So depending on if you have high HDL or low HDL, there's certain genes that are typically related to these levels. So I mean, those genes we looked at initially to try and figure out, why do these people have extremely high or extremely low levels of HDL. But now at this point, since we've kind of exhausted the known gene list, we have to find the unknown genes. So there's a lot going on, and there are a lot of, cholesterol-related genes that do come into play.
So there's 7 that are known, and there are probably more.
Oh yeah, there are tons more. Those were like 7 big players, but there's a lot of secondary players, and there's countless.
So what's something like this going to mean for like our understanding of biology? Because that's really cool that we're starting to get to the point now where it's not like, boom, you have the high LDL gene, and boom, you have the high HDL gene, and that explains everything. It's not like a simple one gene for one kind of trait scenario here. So what's that going to mean, do you think, for biology as a whole, or medicine, years down the road, once you really get a good grasp of genetics?
That's a really great question. So I mean, if we find more of these genetic players, at some point they can be used for screening. So an individual comes in, you want to assess the likelihood of them having really high HDL or really low HDL, and then other kind of cardiovascular consequences from there. So that will be really important as to determining, so this individual, you are probably not going to have a heart attack because you have really high HDL. And so it'll be really helpful for those types of screening and things like that. In terms of, a further understanding of cholesterol and those pathways, I think it'll be really cool, because at this point, I'm hoping I'll find something novel. So if I can, then we'll further expand, the cholesterol pathway and what we've previously defined as, what is known, and hopefully we can expand on that. But I think there's a lot of possibilities around this type of research. So,
and I guess my last question for that is, because I, in my research, study proteins and their modifications. So I like to think sometimes, if my research has any meaning, that there has to be a large environmental component as well. Do you think that the genetics is at least a little bit limited at some point, that there's going to be like a cutoff and we can't necessarily determine what's going to happen in somebody's life based on their genetic makeup, or it's going to get just too complex and we can't figure out all the information? Or do you think we're going to reach a point one day where I'm going to be extinct as a researcher and it's going to be all you?
Well, let me there's a lot of stuff there. I mean, it is so complex that, at this point in time, it's a matter of, figuring out what's environmental and what is genetic. But I think as we move forward, if we can, really figure out how much is environmental, how much is genetic, we can, as, doctors can, go to their patient and give them a really solid kind of lifetime plan if they want to, stay healthy and go from there. So there's, the possibilities, I think I've already said this, are endless for this, since it is so complex. There's so many things going on. But yeah, it's going to be tough to kind of separate the two out and then kind of give a quote-unquote prescription from there for patients. So
awesome. So I want to get into a little bit of the methodologies. You said you have a patient cohort that you're looking at with either a high level of HDL or a low level of this lipoprotein. So what was done to analyze them? How were they assessed?
Well, there's a lot of kind of computer work here, so a lot of bioinformatics and analyses going on. So there was a big, initially all these patients had to be sequenced, so we had to get their DNA sequence just to start, analyzing and figuring out what's going on.
So how do we get their DNA?
Oh, so that one's straightforward. So you can either take a blood extraction or saliva extraction for these particular individuals, and then from there you can isolate the DNA and separate out the cells and the proteins. So that's the straightforward part of this project.
All right. So, and then once you have the DNA, what gets done with it? How do you sequence DNA? Like you mentioned sequencing, and I know we have a sequencing lab here. Sort of what goes into that? What are we doing when we sequence DNA? What are we looking at?
Oh, I know this one. So they get the DNA from the frogs and they fill in the code and then they put it in the ostrich eggs and that's how they make the dinosaurs, right? No?
Maybe in someone else's
Nice
lab.
try. Soon. Yeah. So like, how are you looking at this? So you're taking their DNA, and then where does it go? What's it do?
So the DNA that we get, we're interested in a particular part of the DNA or the genome, so the collective whole. And so we're just looking at actually 1% of the entire genome, and that's just the exome, which are the parts that encode for, proteins and the functional parts of your cell and things like that. And so, for sequencing, we want to amplify this particular area and then from there determine the bases of that sequence. So, is it an A, a G, or T, or a C? And then really we just want to get the letters correct. That's essentially what sequencing is, so we can, get something to work with.
So the patients that you work with, do you personally see these patients?
No, not me. So that was just my supervisor, since he's a clinician. So I kind of do the behind the scenes stuff.
So the reason I ask is because I'm interested in finding out whether they have certain phenotypes. So if they, for example, are overweight, or if they have certain underlying diseases.
Yeah, so we took that into account when selecting our patients to study. So, having high or low HDL can be kind of a secondary, consequence of another kind of dyslipidemic issue or another like lipid phenotype. So we took those into consideration, and we really wanted to make sure that our study population only had some sort of HDL abnormality and not other phenotypes to go along with it.
All right. So now that I understand there's different types of sequencing that you can do, what kind of sequencing do you use, and how does it compare to maybe other kinds that go on out there in the world?
Well, for my project, we utilized whole exome sequencing, and so it's a kind of a subset of next generation sequencing. So because we're looking at such large data sets and we have so many individuals and we want, high quality, when we use next generation sequencing, it's able to sequence millions of DNA sequences at the same time to get a really robust output. So that's what we're going for, or that's the method we used. And then whole exome, like I mentioned, we're just looking at a small fraction of the entire genome because, like I said, these are the parts that have the functionality and are more likely to play some sort of role that we can characterize in phenotype.
You see, like, as an outsider, this is where I find things really fascinating, because, like, from an outsider, right, like, everyone's think that we got genetics and the way that we do genetic stuff really, really down pat. Like, it's in a computer, we can print it and stuff. But then I hear, like, you guys have to take a statistic sampling of all of the nuclei in order to get something. And then I also heard about something the other day where if you want to put a gene into something, you just, like, kind of blast it into them and hope it just ends up in the right place. And it's like, the perception of genetics is so, like, it feels like it's so digital and proper, but it's actually still, like, I'm not saying it's a rough science, but I'm saying it's, like, it's still, like, kind of squishy. I don't know. I don't know where to put it. Genetics comes off as so, like, clean and digital, but it's not.
It would be nice if it was. I mean, I think that's what we want to get at in the future. But I mean, at this, like, in terms of, all the digital stuff and kind of where bioinformatics comes in, we need these high-powered tools to process all of our, huge amounts of sequencing data. So for my project alone, if I just took the raw data, it would be like more than a terabyte's worth of information. And I'm just looking at the exome, which is 1% of the whole genome. So if I looked at everything, I think it was like, I don't know how many terabytes it was. But anyways, so we like to think it's digital because, it has to be digital to analyze everything. But, the issue comes into play when, I'm out of biology, so I don't have like a really hardcore background in statistics or computer science. And so when you go to, bioinformatically analyze these things, you kind of need to know a bit of everything to actually make sense of what you're doing. So that's kind of what I found. I'm like, oh man, I need to, brush up on all these stat things and learn a bit of computer science and programming. So it is definitely digital, and we're getting there. But there are a lot of parts where we're like, okay, we need to kind of streamline this and make it a little more flashy.
So I guess you just mentioned a word a few times that we've spoken about in the past, but what is bioinformatics? So like you've got your DNA sequences, you're looking at it, it's a terabyte or more of information, and then you do something called like a bioinformatic analysis. Like what does that mean?
So bioinformatics, I'd like to think of it as just a kind of a broad term of any sort of, computer processing or analysis that you would do on large data sets. So it takes kind of, a statistical approach and, incorporates all these, fancy computer algorithms to kind of get you to some sort of biological answer or help you get to a biological answer. So bioinformatics, it's a combination of, the biological question and some statistics and a lot of computer science stuff. So it all kind of merges into one, and it's this fancy little method that I have to wrap my head around.
High throughput biological information processing. That's really cool. Like I look at like one little thing at a time, so I've never had an enormous data set that I need to look at. It's all stuff I can do like with a pencil and paper. So it's really cool to hear something like bioinformatics.
Is that like a Punnett square? You do like Punnett squares or something? I know what those are. I took those in high school.
I did those in high school too. I have not used one since high school. Okay, so you've got your DNA, you've analyzed it, and let's say you discover a new gene that's a little different in one patient than in the rest of them, and this patient happened to have insanely high, or I guess one group of patients happened to have this similar mutation, and they have really high HDL or LDL. I guess HDL for you. What would you do next? Like, how do you have to characterize the gene? Like, what would you be doing?
So there's a couple of things that you would need to do. So first, with next generation sequencing, there's potential for, sequencing artifacts or technical artifacts. So one of the first things you want to do is actually make sure that what you think is interesting is real and not, just a mistake. So you would go back to kind of the old school gold standard of sequencing, which is Sanger sequencing, to actually make sure that what you're looking at is, real. And then from there, you need to create a story for why the gene you're looking at is actually related to, HDL cholesterol. And so you'd move to functional studies, and I'd actually get behind a lab bench instead of a computer and put on the lab coat and do functional experiments to, see does this particular gene or mutation actually relate to HDL and change HDL levels. So there's a big process after the bioinformatics section. So.
You just lit up when you said put on a lab coat and get behind the bench. Are you like really looking forward to doing that?
Yes. It's really hard to, sit at a computer and try and sort through, terabyte of data to find something interesting. So it would be a good change of pace.
Yeah, it can be a lot of fun. All right. So what do you hope to get out of, I guess, your master's research? Not just you, but like, are you hoping that you're going to find a gene that's really going to make an impact on patients' lives? Or
it would be unreal if I could find something, that big. Like usually people might find, a one variant or one mutation that could help explain the HDL pathway. So if I was lucky enough to find like a whole gene and somehow relate it back to HDL, that would be incredible. But I mean, if I can just contribute a little bit with, one mutation or whatever, that would, be really satisfying to know that all the work that I've done kind of helped, uncover the mystery a little bit. So if that would be a really ideal outcome. And, I'm learning a lot along the way. So even that already has been super amazing.
So from there, I guess what I want to ask is, what is an increased understanding in HDL and LDL going to do for cardiovascular disease as a whole?
Well, if we can get a better insight into all of the players for HDL cholesterol and LDL cholesterol, that would really help regarding cardiovascular disease risk, because you're depending on the level of, your good or bad cholesterol, you could have very different outcomes for, a heart attack and things like that. So the more we understand about, cholesterol and these types of, players, the more that we can do for cardiovascular disease research and those types of things, because I believe cardiovascular disease causes 1/3 of deaths in Canadians. So that's like, a huge number. So the more we can figure out to kind of help, kind of combat this issue would, it just makes sense to me.
So it causes 1/3 of all deaths?
In Canada.
How many non-deaths does it cause too? Like on top of the third of all deaths, like I guess, one in how many people like want to?
I'm not sure off the top of my head. I should know that. This is why I should know this already, but
You will know this after this. Anyway, so it's really common. I guess when we talk about cardiovascular disease, just to go more broad now, what kinds of diseases are we talking about? Are we just talking about heart attack? Are we talking about heart attack and stroke? Like what, I guess, are the cardiovascular diseases that exist?
Well, you already said two of them, and I mean,
Yes. Nailed it.
and atherosclerosis and hypertension. There's so many things that even I still haven't fully comprehended yet in my week and a bit of being an official master's student. But there are so many kind of offshoots of cardiovascular disease, and so many areas that are involved, I guess.
So we did focus on cardiovascular disease. Does cholesterol cause any other types of diseases?
There are actually a lot of diseases associated with your lipid levels, and so not necessarily cardiovascular diseases, but their names are super fancy, and I wish I could explain them. But there's a lot where your fat deposits, they're not in normal spaces. So I know there's one, you get a lot of fat buildup and deposits on your liver. So obviously that's quite an issue. But there are lots of other diseases related to lipids that people don't really know about because they're not on the front page of the newspaper all the time. But there are a lot of things, and these individuals have to go through a lot and take lots of medications just to kind of control their lipid levels. So I mean, there is a lot kind of within the lipid cardiovascular research world.
So do you think that every person should be getting their HDL or LDL checked on a regular basis? Because what I'm worried about right now is that I'm not a large person or anything, but I would be concerned that my HDL or my LDL could be off because I'm a grad student and I don't eat particularly well. So
well, I mean, I think the majority of the population has relatively normal levels of cholesterol, and if you eat well and you exercise, that'll help. But of course, I think you're thinking of extreme cases, but I think you're going to be okay. And in terms of your genetics, if you go get screened, then you can determine it. But for the majority of the population, it's pretty environmentally based.
Okay. I guess for my very last question, we have 1 minute left. Tristan's making a face, but you just mentioned genetic screening. So something like 23andMe, is that an okay way to get screened, or would I have to go to a real genetic counselor?
23andMe doesn't look at lipid traits as I recall. So, but if you really want to know, I would go to a physician. 23andMe is kind of like a fun, let it go, let's go see what it is. But I wouldn't put any, I wouldn't bet everything on 23andMe. I'd go see a doctor if you really want to know.
Okay. Well, I think that's all the time we have. So thank you so much for coming on the show. I know it's early on in your master's and you were a little bit nervous about it, but you did an amazing job. Thank you very much. Hopefully you'll be able to come on the show another time later down the road. Everybody, this has been Gradcast. I'm Alex Moszczynski. See you guys all next week. Thank you very much.
Yup. That's all we got for this week. If you like this episode, share it with someone. Check us all out on Twitter and Facebook. Both you can find through Gradcast Radio. You can go to our website to see more episodes at gradcastradio.ca. And if you want to come on the show and talk about your own research, great line for your CV, go to gradcastradio@gmail.com. The theme is Happy Boy by Kevin MacLeod, and we will see you guys next time.